Welcome, folks, to the lecture on Chapter 13. So far in this class, we've introduced microbiology, some of the history around microbiology, some of the tools and techniques that we're going to be using in this class and in microbiology generally. We've talked about prokaryotes and some examples of prokaryotes, although we will get back to that at the end of the term and talk more about specific organisms that are relevant to various medicine, environmental, microbiome, so on and so forth. We also talked about the cell structure of prokaryotes. And then today, we're going to be talking about viruses and prions. We'll spend most of the time talking about viruses. We'll also talk about viroids and prions as well, all different types of infectious agents that are non-cellular or non-living, but they have some characteristics that allow them to interact with and interface with living systems. We know that these are very relevant because we live in a time that is a pandemic that's related specifically to viruses. Okay, so I do want you to pause this video, review any material or content that from previous lectures that you want to go over. Also, go into the first link in the video description. There's just a one-sheet review of viruses and who they are from a virology book that I really love that allows us to just get an overview of who it is that we're talking about. Okay, so go ahead and do that and jump right back into the video. Okay, folks, welcome back. So we said that viruses are non-cellular. The organisms that we've talked about so far are organisms. They are in fact alive, and they have those characteristics of life that we had mentioned previously in this class. And so specifically we focused on prokaryotes like bacteria and archaea, although archaea do not cause infections in humans that we know of, but bacteria do. Now, there are agents of infection or other agents that are out there that are what we call acellular or non-living. And these includes viruses, viroids, and prions, and that's the focus of our lecture today. And so even though these organisms, they do not have the same full-on living machinery as prokaryotes, they do have similar machinery in some ways. And you had kind of interacted with that in one of our previous lectures where we did a discussion on viruses and the characteristics of life. And it is those similarities that actually allow for viruses, viroids, and prions to interface with living systems and cause infections and become relevant and cause disease. And so we're going to talk about those various categories and some of those characteristics. So what is a virus, right? A virus essentially is a combination of some genetic material, okay? That genetic material might be DNA or RNA, might be single-stranded or double-stranded, but it's some sort of genetic material that's then protected somehow. Usually there's a capsid involved. Sometimes there's other layers of protection like an envelope that's involved, but that entity is called a virion, and that virion is able to infect cells of various hosts. And in fact, we know that viruses infect all living organisms in all kingdoms, from archaea, bacteria, fungi, plants, animals, and protists. All are infected by viruses. The word virus comes from the Latin for poison because it was early on kind of recognized that these were causing infection and disease, although we didn't know historically exactly who they were. And eventually there's some research on the tobacco mosaic virus where in like the early 1800s, where we had a scientist who literally was trying to figure out what was causing this infection on a tobacco plant, and he filtered out the sap off of an infected tobacco plant, and with a filter that would catch any living organism, including bacteria, and still that sap was causing infection and disease. So he's saying, okay, then there is a sub kind of cellular or a smaller than cellular particle that is causing this tobacco mosaic infection. But we still didn't even characterize that specific virus until the 1930s, right? So this research has kind of developed over microbiological history to kind of help us understand, like, who are these very small agents that are right at the cusp of living or non-living, but they're still able to cause disease. When we say very small, what does that mean, right? That means that there are some viruses that 100 million of them, 100 million of them would fit across a period on a page in your textbook, like the punctuation period on a page in your textbook, right? There are some, of course, that are larger than that, even the size of a bacterium, so about 1 micrometer. So it does fluctuate, does vary, but these are very, very small particles indeed. A useful analogy is actually computer viruses, right? So computer viruses, they have characteristics. They have, essentially, they're a file, right, that has some sort of code in them that can cause it can cause infection, can cause changes in your actual software. But that file in and of itself doesn't provide a function or doesn't do any specific function. But once it has been let loose into your software, then it can actually create domino effects and make changes in your software and sometimes other folks' software that are on your network too. And so in that way, it's a very simple kind of unit. In this case, it's a unit of a computer file or code that is then able to impact the larger system once it's introduced. And so these viruses that create infection and disease are very similar to computer viruses. Now, they're this very simple unit. On their own, they're pretty much useless, but then once they actually are let into the larger unit, they have similarities enough that they can communicate with and cause domino effects within that broader system. So be that the human body, for example, or be that your computer software or your network software, for example. Okay. So let's talk a little bit more specifically about the kind of anatomy of a virus. So the infectious agent itself is called a virion, and it at least needs to have some sort of genetic information and some sort of protection around that. That's a capsid. It can be a very simple capsid, or you can actually have capsid in layers of other things like envelopes. Envelopes are similar to membranes, although they don't have the same functions. So if you just that unit that has the genetic information and the capsid is called a nucleocapsid. And then again, sometimes there are layers beyond that again protects the virus. Now, the virus, essentially, the functional part of it is the genetic information. So the goal of that particle is somehow, usually it's just random occurrence, that it finds cells. Those cells are usually, those host cells are very specific organisms. Sometimes there are even very specific cell types within organisms, although there are a couple of exceptions of generalist viruses, but oftentimes viruses are very, very specific. And they use that capsid or the envelope to then, be recognized by the cell, connect with the cell, and then entering into the cell. Their genetic information, the genetic information being the kind of functional part. And when I say recognized by the cell, I don't mean that the cell recognizes it as a virus, because if it does, then it usually has defenses against it. But when I say recognition, I mean just attachment via proteins or lipids that are kind of communicating with one another that allow that virus to actually attach to the cell. Oftentimes, pretending to be the host itself or kind of evading the host's defenses. Okay. So capsids can be this really beautiful structure, actually. One of the most common, there are different structures. You can have a helical structure, I'll show you in a minute, but one of the most common structures is what's called an icosahedron, and these are a 20-faced geometric shape, and all you have to do to add size to this geometric shape is then to add the subunits of proteins that make up these capsids, and that allows you to expand in size or shrink in size, but maintain a very kind of stable structure. This icosahedral is very stable. And so it's also just a very beautiful structure. You can kind of see here, you can see the genetic information inside and then the capsid on the outside. We'll talk about this a little bit more, but there's levels of kind of complexity of viruses. Some are very simple. They just have the genetic information with proteins wrapped around that information in just kind of a helical structure. But some actually have tail fibers and kind of all of these different filaments that allow them to attach to their host cell. And they have these really, really complex, but also very just kind of intriguing and beautiful structures as well, like this T4 bacteriophage here. So we'll get back to shape in a sec, but in terms of viruses, kind of the basis for understanding which virus you're working with is to understand what kind of genetic information it has. That genetic information, again, is really the functional piece. The capsid or the envelope is essentially just to kind of transport it, get it to where it needs to go. But the functional piece is that genetic information inside. And so this is how we actually categorize viruses, is that we have viruses that might be single-stranded or double-stranded. They might be RNA or DNA. They might be circular or they might be linear. And understanding which is which gives us an idea of which virus that we're looking at. Now, go back to the central dogma of biology, and you're going to remember kind of, okay, remove yourself from the viral world and put yourself into the eukaryotic world for a second, and think about how we go from the DNA that's inside of the cell. This is the same for bacteria and prokaryotes as well, just, without the membranes. You have the DNA that's inside of the cell. That's your blueprint. And then you make copies that are single-stranded RNA. And then that single-stranded RNA is then converted into proteins. It's read and via ribosomes and converted into protein. So from a viral perspective, whether they're double-stranded, so if they're double-stranded DNA just like the host, then they tend to enter into kind of generating their proteins when they hijack the machinery of the host at that initial phase, where they kind of look similar to the DNA of the host, right? And then they are able to go through those processes and then generate their own proteins by hijacking the machinery of the host. But if they come in as a single-stranded RNA molecule, then they kind of already look like a copy. And then they'll just jump in at this portion where they just need to translate into proteins, right? Now, what's interesting is we don't really see double-stranded RNA in hosts, and we don't really see single-stranded DNA in hosts, but we do see those in viruses, right? And then that allows them to essentially hijack this process in various areas where it looks like just a single strand that's replicating of the DNA, for example, or there's a double strand of RNA that's able to be separated, and then one is used for a template for generating proteins and one is used for a template for generating more genetic information. And so it's really interesting that the diversity of genetic information in viruses is actually more than the diversity that we see in genetic information in terms of kind of those kind of structural units and how they look, double-stranded, single-stranded RNA, DNA in the hosts, right? Now, lots of variety in terms of kind of what that genetic information looks like. But generally speaking, viruses have really, really small genomes, okay? So just to give you a comparison, the human genome has between 20 and 25,000 protein coding genes, okay? The smallest bacteria has about 1,000 protein coding genes, 1,000. Viruses, on the other hand, usually have just a handful of protein coding genes in their whole genome, right? 2, 3, 5. The largest one that we know of, the largest virus that we know of, which is a megavirus, has 200. Okay, so that's atypical. That's kind of really large for a virus, 200, but generally speaking, it's about just enough to count on your hand in terms of protein coding genes. So these are very simple genes. They just essentially have genomes. They just essentially have the genes that they need to replicate their DNA, generate whatever capsid proteins that they need, and then assemble those once they hijack the host. But they're still able to wreak havoc, right? So here you can see this is essentially an E. coli cell that has just kind of been popped, and then genetic information is kind of extruded into its environment. What you're looking at is actually not all of the genetic information of the E. coli bacterium, just some of it. And then this circular genetic information genome right here, that's very small in comparison, is the viral genome. Okay, so it gives you kind of a visual comparison in the size of these. Interestingly enough, when we talk about viroids, they don't have any protein coding genes. They just have like a couple hundred base pairs, and they're still able to cause disease, particularly in plants. They're still able to cause disease, and we'll talk a little bit about that later. Okay. But it's just that foundational kind of point that the genetic information it matches, right? It's a similar kind of unit that they're using in terms of DNA or RNA, and that's how they're able to wreak havoc. Hosts. So I think we mentioned this before, but the hosts of viruses tend to be very specific because the proteins that are on the outside of that virus are very specific in terms of recognition. There are exceptions to this, like the West Nile virus, which is actually a generalist and can infect various types of organisms. But generally speaking, viruses tend to be pretty particular in terms of who they infect. But they do infect all of the 6 kingdoms, as we had mentioned before. All organisms have some sort of association with a virus that can infect them. So examples, you can see this tobacco plant with a viral infection. Here you can see a bacteria, and these are those T4 bacteriophages that we had talked about, the icosahedral head, and then the tail and filaments. It's very interesting. And this last one is HIV infecting a white blood cell. So just in terms of terminology and word dissections, whenever you see the suffix phage, Phage, that tends to refer to a virus. So bacteriophages are viruses that infect bacteria. There's not an exact equivalent for animals, for example. We just call all infectious agents of animals zoonotic infections, zoo meaning animal. But when you do see the term phage, it's generally speaking that you're talking about a virus. Okay. Another note, most of the viruses that most studied viruses are your bacterial viruses and your animal viruses. So what we'll do is actually we'll focus on bacterial viruses in this lecture. There is a discussion of animal viruses. We're just going to have a couple of notes in lecture, but do read over that part. And then just know that there are some differences for like fungal and plant viruses, although we just haven't studied fungal and plant viruses quite as much as we have bacterial and animal viruses. One of the interesting things about fungal viruses is because they have this thick cell wall, plants as well. It does mean that viruses tend to be intracellular instead of external to the cell, and tend to be shared once those cells replicate or connect with other cells rather than externally. So that's just kind of one unique difference. Okay, so let's visualize a little bit the size comparison of viruses. So you're looking up here, we're going to want to start in this square right here, where we're looking at a red blood cell. Red blood cell is about the size, is about a 10 micrometers in width, right, in diameter. And so this is something that we can see pretty easily with the microscopes that we have in lab, for example. And so that's our red blood cell. Then beside of it, you have an E. coli bacterium, which is about 1 micrometer in length. And so we need to be able to see E. coli well in the lab, we need about 1,000 times magnification, right? 1,000 times magnification. And still, essentially, we're looking at rods, right? It's still difficult to see the detail within that bacterium. And then, even smaller yet, instead of talking in micrometers, we're talking nanometers, are various viruses that we have here, from smallpox to bacteriophages, so on and so forth, right? So these are very, very small particles. In fact, there are viruses that seem to actually infect other viruses. So even very, very small viruses that are actually infecting these very small viruses. So even like smaller and smaller yet. Okay. So we talked a little bit about the genetic information that's within viruses, can be diverse in kind of how it's kind of formulated. Now let's talk a little bit about capsids. Capsids are essentially a protein structure that's made up of capsomeres. Capsomeres are, protein specific for forming capsids. We had talked about this a little bit, that essentially the capsid is there to protect the genetic information. And I want to show you an image because we kind of have these 3 categories: helical, polyhedral, so the icosahedral capsid that I showed you before would be in that category, and then complex. Also, the one that I had showed you before is complex as well. And so you can see that here. For the helical capsid, it's just a helix of those capsomere proteins that is just protecting the genetic information. Very simple. Then that second, so that first image right there that you're looking at, that's a common cold virus. Then you can have a little bit more complexity where the capsid actually takes on one of those polyhedral shapes. There are different shapes that can be taken on, but one of the most common, because it's so stable, is that 20-faced icosahedron that we had talked about before. And that 20-faced shape can take a larger or smaller kind of size. And then that protects the genetic information, which is then inside. So this second image here, this first image here was the tobacco mosaic virus, and the second image here is the common cold. And then here you can add layers of complexity where you have some other structures like an envelope, or you have other kind of structures on the outside that give it more protection or more ability to interact with the host. And these tend to be common for animal viruses, specifically because they're interacting with that membrane of the animal cell. And so they tend to have what's called an envelope, not always, but they tend to have an envelope that is actually made off of the host's membrane so that it looks similar to the host, and it can actually trick the immune system of the host into thinking that it's self. So here we have rabies, which is an animal virus, okay, in this third image here. And there are other complexities. So here you can see the head, which is similar to just the capsid on some viruses, would just not have this bottom part right here, this whole tail and all of that structure. That's that icosahedral shape. But then sometimes there's extra complexity on there where you actually have these tail and filaments that actually connect. So you have initial, in this case, with the T4 bacteriophage, initial recognition actually occurs at the tips of these, what look like legs, but they're called tails. And then once recognition happens, this base plate right here then connects again with the bacteria, because these infect bacteria, connects. And then this part right here actually works just like, kind of like it compresses, kind of like a helix or like a spring. It compresses, and then that allows for the genetic information to be released through this, which is actually a hollow tube. Okay. So this is just one of the most fun looking viruses. So you see it often, but actually oftentimes what you'll see are much simpler. So a virus that just wouldn't have this part, for example, right, would just be this part, and that would just be one of your, viruses that has the genetic information and the capsid, for example, right? And it wouldn't have all of these extra kind of recognition points and so on and so forth. So it just depends on the virus that you're looking at. Some of them have simpler structures, some of them have more complex structures. Okay, so we said that all viruses are going to have the genetic information and the capsid. Okay. Then you have some unique viruses that have another layer on that, which is the envelope. These tend to be, like we said, animal viruses. And essentially, as the viruses, the virions, and you'll see this when we talk about the lytic and lysogenic cycles, as those virions are released from the cell, they actually pick up some of that membrane from the cell itself, and that allows it to be recognized as self. Now, that is a huge plus, that these virions kind of exist within the body and are able to trick the host immune system into accepting them and allowing them in the presence of the cells that they then infect. But the fact that it's a lipid bilayer, that just, by definition, that lipid membrane tends to be a little bit more fluid, as we had seen in the last lecture, and it can be susceptible to desiccation, drying out, so on and so forth, detergents, chemicals that are used by the host to defend itself. So it does mean that those viruses are a little bit more vulnerable, but they do have the added benefit of being recognized as self. So it's always a trade-off, right? Whereas what we call naked viruses or virions, do not have an envelope. They're a little bit more susceptible to wear and tear in harsh environments because they don't have that lipid bilayer. Instead, they have the capsomeres, which are those proteins that form the capsid, and those just tend to be more stable than the envelope that's made out of a lipid bilayer. And so, living systems or pseudo-living systems in this case are always, some sort of trade-off that you see there. And then each virus is just going to kind of be most optimal in terms of infection of its host. And of course, ever evolving, as we see with having to get updated boosters and shots year by year for various viral infections. Okay. And so here you can see this is a helical capsid that's protecting the genetic information, but here you have the membrane, the membrane-like structure that has proteins, glycoproteins on it, and so this membrane-like structure is called the envelope. It doesn't form this. It doesn't have the same functions of an actual animal membrane. cell, right? It doesn't do, like active transport and so on and so forth, endocytosis, exocytosis. It's literally just a kind of a guise, like a disguise, right? It's not a functional membrane, and that's why it's not called a membrane. It's called an envelope, even though it's made from very similar components. And then what happens is when that virion comes out of its host, is being released from its host, and it picks up that envelope from the host, it does also pick up proteins from the host, but oftentimes the virus also encodes surface proteins that are unique to it as well. And so that envelope has proteins that are both from the host membrane as well as unique to the virus. So it usually has a combination of proteins there. Okay, so you have some summary tables in your textbook that allow you to kind of review that material. And I would welcome you pausing the video and reviewing where we are so far. Okay, so in terms of phylogeny, right? Virus phylogeny or classification. Now, we already said that the most important kind of component for deciding which virus you're looking at is the genetic information, and then also the structure of that virus. And indeed, viruses are categorized into families and groups similar to living organisms, like bacteria, archaea, eukarya. And there are some researchers that consider viruses a fourth domain, right? Bacteria, archaea, eukarya, viruses. But generally speaking, researchers and scientists consider viruses non-living components or acellular components that do have some living characteristics, right? And so even though the naming system is similar, this is to allow us to classify viruses, but generally speaking, we still consider them acellular or non-living. Now, you don't need to know the families of viruses, but do know that there's a similar naming system. Oftentimes, the naming system is related to, a predominant virus that's in that group. So, for example, Habadnoviruses or Habadnaviridae is a group of viruses that also includes, the virus that causes genital herpes. And so it's kind of that whole group is named by that. You also have Picornaviridae, and I just, want to remind you about dissecting words, pico meaning small, and so some of the smallest viruses are in this category as well. And so you can see kind of different categories with the different viruses within them. Now, we will go back to specific viruses at the end of the term and actually interact with some predominant viruses that cause disease and wreak havoc. And so we will go back to, these families here, but you won't have to know them for this chapter. Okay. So we know what a virus looks like now. We know its components. We know, a little bit about how it interacts with or how it interfaces with living systems. And so this gets us to that contention point, right? That viruses generally are considered acellular or non-living. So how is it that they do their work, right? And so viruses are what we call obligate intracellular parasites. There are other obligate intracellular parasites like bacteria, like protists that are parasites, right? But viruses, they do act different within their host. They do act differently within their host. And the really key difference between viruses as obligate intracellular parasites versus a bacterium or, a protozoa that's a parasite is that living parasites, they remain intact. They have their own machinery for replicating themselves from the parent to the daughter, so on and so forth. Whereas viruses, no, they actually once they enter the cell or upon entry of the cell, they dissociate, and that genetic information hijacks the host's machinery because they don't have their own machinery for replication, for protein generation, so on and so forth, right? And so viruses need all of these extra things from their host. They're not just hanging out inside of the host and replicating, they're actually taking over the cell of the host and using the machinery that they don't have of their own, right? And so, some of the things that viruses specifically need is they need the enzymatic systems that are provided by the host that allow them to generate biological macromolecules. They can't generate them on their own. They need the enzymatic systems that provide them the energy to do so, to generate those macromolecules and assemble themselves. They need the ribosomes. They don't have their own ribosomes. All other, like any living system is going to have ribosomes to actually translate and code into DNA, into proteins. And they also tend to need the membrane components that allow for ion transport and assembly of various virions that then are going to be released outside of the cell. So they really have all of these extra needs. They're really going into the cell almost without anything, without any tools, and they're hijacking that cell. They're taking over that cell, and they're using its machinery. And so upon entry into the cell, viruses kind of take two replication phases, right? Either a lytic phase. Lytic, what is the word lytic? Or is to explode, a lytic phase, or a lysogenic phase, which is kind of a hiding phase. And so here you can see just a cartoon image of a lytic phase here. Essentially what's happening is the virus is inserting its genetic information and then hijacking the cell immediately upon entry and then generating more of itself. And that has these various stages where you have attachment, entry, synthesis, assembly, and release. And so you can see that here.
Viruses are agents capable of infecting cells and reproducing, but it can be argued whether a virus is alive or just a complex group of molecules, since it requires a host cell in order to reproduce. Whether they are alive or not, they're able to co-opt the host cell's machinery in order to do the bidding of the virus, the replication of the genetic material of the virus, as well as the structural elements that package and protect the genetic materials and enable the virus to infect new host cells. There are many different types of viruses, infecting a large range of host cells. They all follow the same general life cycle, with variations in the details of how each accomplishes its replication.
So essentially what you're looking at with the lytic phase is you have attachment and release of the genetic information or entry of the virus into the cell, and immediately those virions are then replicated and produced. Now, for some viruses, this whole process, all of these steps takes like 25 minutes, right? So it can be, it's a pretty much immediate process. The virus, the virion enters the cell, infects the cell, takes over the cell. Oftentimes, the virus, upon entry into the cell, will actually disintegrate the host genome, genetic information, disintegrate that information, and then the machinery of the cell is then left to generate virions. Okay, so this is kind of the process that you had seen in the video. A couple of important terms here. So you have entry, and you can see there's one virion that enters, but then it disassembles. So, you have kind of the genetic information that is just there. And then you have this exponential increase in the number of virions that are produced. And then at one point, the cell doesn't have the capacity to contain those virions anymore, and they're released. And so that's called the burst size. And then, the burst size, and then the time at which it takes, or how much time it takes for that to occur, is the burst time. Now, T4 bacteriophage, which you had just seen in this previous image here, produces about 100 to 200 virions in that 25 minutes and then bursts out of the cell. And it just depends on the cell, the host, the virus that we're talking about. But it can be a very quick process and produce a really large viral load. Okay. In the lab, it's actually pretty easy to see the lytic cycle. Essentially, what you do is you plate a bacterial lawn. So what you've seen so far in lab is a streaking method, either with a sterile swab or inoculated with a sterile swab, or with a loop. But in this case, we would just make a full-on lawn by spreading a drop of liquid that has the bacteria and the viruses mixed together. When you spread that over the plate, you get a consistent coverage of that plate, and that allows you to see, once those bacteria grow, you can actually see where the viruses are, are killing out the bacteria in that area. Now, this is when you have a virus that immediately comes into a cell and replicates. That's not always the case, though. Viruses can also enter a cell and then just kind of hang out and wait. And this allows them to do two things. One is, essentially evade or hide within the cell. And two, it actually allows the virus to be replicated via the replication of the host. Okay, and so what happens is you have the insertion of the viral DNA, but not just into the cell, but actually into the genome of the host, right? And so this yellow piece here is the viral DNA. And these, again, will be DNA viruses, right? Characteristically, they will have a double-stranded DNA that allows them to insert into, the host genome, generally speaking. And they will insert in there, and they will actually produce various proteins and chemicals that inhibit recognition from the host, and also even inhibit further viral infection from other viruses as well. And they just kind of exist within that cell. Then as that one cell replicates once, then twice, then 3 times, then 4 times, and continues to replicate, they can actually hang out in the cells for generations. And then they are essentially being replicated via the replication of the host, instead of 10 viruses having to infect 10 cells or 100 cells or 1,000 cells and 1,000 viruses. It's just one virion that infects the initial host and then is replicated and replicated and replicated and replicated. And then some sort of chemical induction basically induces that virus to then, that viral genome, to jump out of the host genome and move into the lytic cycle, which we had talked about before. Okay, so you can essentially go straight into the lytic cycle, or take a pause and go to the lysogenic cycle and then move into the lytic cycle. And different viruses tend to take on different cycles, right? So you might have these viruses that are able to just kind of insert into the host genome and kind of hang out there. You might have viruses that just tend to go straight into the lytic cycle without taking a pause to go into the lysogenic cycle. Okay. Yeah. So I think we had mentioned this already, essentially, that the viral genome, instead of disintegrating the host genome, it's actually utilizing the host genome by by combining its genome with the host, which we had mentioned. Okay. And these are some viruses that do that. And you can see that kind of system here, where the initial viral infection occurs here, and it could it could move into the lytic phase, or it could move into the lysogenic phase, and continue to be reproduced by the cell, and then eventually then move into, based on some chemical induction or some environmental induction, then move into the lytic phase. Okay. There are viruses within our bodies that can do this. And in fact, the human genome has about 8 to 10% of it that's remnant of viral genome. That's a remnants of viral genetic information. So it's kind of interesting to know that, essentially we're part virus in a way. There are also viruses that are able to move between a lytic phase and a latent phase, where they kind of hide or they infect other cells in the body. And so this is the case for herpes that produces cold sores. Once you have that initial infection, then sometimes it'll go away and continue to come back, because it kind of exists in a latent phase in some other cells in your body, and then comes back into the kind of infectious or lytic phase in your epithelial cells as well. So there's kind of, this can look different in different ways and have various implications. Okay. So animal viruses. We said animal viruses are very similar in a lot of these ways, but that one difference that you tend to have an envelope on an animal virus makes it where that entry is a little bit different. The envelope is similar to the membrane, and so entry looks a little bit more like endocytosis. If you remember from membrane function, then it does look like that kind of initial stages of viral entry in some of bacteriophages and things like that. And so that's that benefit of the envelope, is that it's able to kind of recognize as self on the host. The host recognizes it as self and then incorporates it, just like it would incorporate through endocytosis something from its environment, nutrients or so on and so forth. There are a couple of other differences of animal viruses. I'll let you read about those, but that's the primary one. Okay. So that's essentially viruses, right? And we can go back to this question, but we had already, discussed it in our previous class, in our previous lecture. Is, are viruses alive? And essentially what we've seen here is that viruses really do interact with, they hijack a living interface, and then they're able to wreak havoc and cause problems. But they themselves, in their own unit, are not a functional living system, right? I will also put a review video in the description below on viruses. Now, it just is a general review via the Amoeba Sisters, but what I really want you to think about when you're reviewing this video is to think about your project, your term project, and how you do have the option to generate a graphic novel. Because when they talk about the lytic and lysogenic cycles, those are kind of perfect examples of how that could be used to generate a graphic novel for the class. So it serves as both a review and also kind of a reminder of this term project that's coming up, and starting to think about some of the options that you have in terms of generating that project. Okay. The other thing that I want you to do is jump into the Jamboard that is in the link below, and talk about or give information about coronavirus, COVID-19. What are its characteristics? Does it have an envelope? What is the structure of its capsid? What kind of DNA does it have? So on and so forth. And give that information about coronavirus, which is really kind of integral to our lives these days and related to this discussion. Okay, so I can let you kind of pause the recording here and then jump into this discussions of, then we'll jump into discussions of viroids and prions. I don't think this lecture is going to end up being two separate parts, just FYI. So go ahead and pause, review your notes on viruses, and we'll be right back. Okay, welcome back. So let's talk about viroids. I think we had mentioned viroids a little bit already, but essentially these are just, very small fragments of genetic information, just a couple of 100 base pairs long, and they tend to be circular, they tend to be single-stranded RNA, so just like the most simple, most basic genetic information, and they do not code for proteins. So instead, the way that they actually cause infection is that RNA connects up with the mRNA of the host, and then the host detects it as other, or kind of it starts essentially disintegrating its own mRNA because of the presence of that foreign RNA, single-stranded RNA. And it's that actually stunts the growth or causes the infection. So even though they don't even code for proteins, they're still able to generate disease. And this tends to be in plants, although there are some other viroids as well. And so you can just see how small the viroid genetic information is. And even though it's so small and it's so simple, and kind of it's the way that it impacts the host is by the host then degrading its own RNA, it does inhibit growth and it does cause disease. So you can see this in this potato viroid and how it's inhibiting the growth of the plant and the potato. Okay. Then the other kind of small infectious agents that we'll talk about in this class are prions, okay? Prion is actually kind of like a shortened name for proteinaceous infectious agent. And so what we want to review here or remember how proteins are formed, okay? So the protein, as we had remember back to the dogma, the biological dogma, central dogma, is that you have the genetic information that's encoded in the DNA, and then a copy of that genetic information that comes out as RNA, and then that RNA via a ribosome is translated into a protein. Now, that the synthesis of the protein doesn't stop there though, because the protein is actually functional via its shape. And so there has to be specific folding and shaping of that protein for that protein to be functional, okay? Now, prion diseases come into play where in proteins that are important in terms of, neurological function that have actually two stable folding shapes. And so essentially one of those is the correct folding of that protein. And if you remember again, back to biological molecules, the folding of a protein is based upon, the hydrophobic hydrophilic interactions and various interactions within that strand of amino acids that then allows it to fold in on itself and make a 3D structure. Now, in some of these proteins that are important for neurological function, there's actually two stable shapes, the one that is the correct function and the one that is actually incorrect and useless in terms of its function. Now, the problem occurs, and so I can show you these. These are these PRPs that exist on the membranes of cells within, organisms' brains, right? Animals' brains. And so the normal folding is this one that you see here, whereas the mutant folding that's in some prion diseases is what you see here. Now, you might say, okay, well, it's folded incorrectly. Doesn't the cell have mechanisms to just get rid of those? Yes, indeed. When proteins are folded incorrectly, you have chaperone proteins and lysosomes and things like that it essentially kind of collect those proteins and then can discard them. The problem with proteins that generate prion infections is that they are actually able to create a domino effect where the misfolded protein, because of its orientation in terms of those features of the different types of hydrogen bonds and so on and so forth, actually leads to the misfolding of normally folded proteins around it. And it creates this domino effect. And so this protein here, because of the way it's oriented and because of some of those charges on its surface, it actually misfolds the adjacent protein, so on and so forth. And that creates a full-on domino effect where all of the proteins that are these PRP membrane proteins in that region then become misfolded. Okay. That gets us actually to the name cow disease, which is an example of one of these prion disorders. The scientific name for it is actually bovine spongiform encephalopathy, and that spongiform is referring to, I'm going to show you here. Essentially what happens is as those prions create those domino effects, they actually create these regions of misfolded proteins, and the body recognizes that. As we had just talked about mechanisms for the body to recognize various proteins that are misfolded, it recognizes that and it starts to destroy those regions, right? And as it destroys, I mean, the impact is it's so large that you can actually see it visually under the microscope as like holes, like a sponge, right? Because all of those areas have actually been destroyed or removed by the body. Eventually, when that happens enough within the brain tissue then no longer can function correctly and appropriately. And that's where you get the severe symptoms and even death that are related with some of these prion disorders. Okay, so we can just see that here.
In 1982, Stanley Prusner discovered that a sheep disease called scrapie was caused by a proteinaceous infectious agent, an infectious agent composed entirely of protein without any nucleic acid. Prusner named these new agents prions for proteinaceous infectious particles. Prions do not reproduce like other infectious agents by multiplying or growing. Instead, they cause other normal cellular proteins of the same type to misfold into the infectious form. For his work, Prusner was awarded the Nobel Prize in Physiology or Medicine in 1997. Since 1982, several other diseases have been shown to be caused by prions, including bovine spongiform encephalopathy, also known as mad cow disease.
So initially, when scientists began to study these prion disorders, there's a lot of pushback because, I mean, we've already seen with viruses that it can be kind of, it's interesting that an acellular particle can actually cause disease. And so that these non-living particles were causing such significant disease was there was pushback against that. But we know that this is a one way in which disease can be caused via these acellular and completely non-living particles, which are prions. Okay. So you can see there are a number of human diseases as well as animal diseases that are known to be caused by prions. Also, there's some preliminary research that suggests that Parkinson's, Alzheimer's, ALS may be related to prions and misfolding of proteins, although that research is still ongoing, so it's not necessarily clear about that. And so essentially, those are acellular infectious particles, viruses, viroids, and prions. And so in this lecture, we've talked about viruses in detail, and we've focused on bacterial viruses because that's where a lot of the research is, but we also talked about animal viruses a little bit, and you're going to read on that further. Viroids, we talked about viroids, which are very, very simple. They're just kind of like the simplest piece of a virus that is not even coding for proteins, but it's still able to generate infection and disease, and also prions, which is just a disease of the misfolding of proteins. And so all of these are essential to kind of understand because they're important in medicine and disease. And so that's that for this chapter. You do have this table in which you have comparison between viruses, prions, and prions and viroids. So I will see you in the next set of videos, and thanks for listening.